কি এটা হচ্ছে?
ওই
হচ্ছে স্যার,
ওম সত্যম আর রাতুলের সাউন্ড অন করা আছে, ওই জন্য ব্যাকগ্রাউন্ড ঝামেলাগুলো আসছে।
দেখছি স্যার। রাতুল, mute your audio. ওকে স্যার, স্ট্রিমিং হ্যাজ স্টার্টেড, আমি আপনাকে হোস্ট করছি।
ওকে। ওকে, আমার স্ক্রিনটা কি শেয়ার হয়েছে, দেখা যাচ্ছে?
হ্যাঁ স্যার।
Okay. Okay, good afternoon, students. Let us continue with our pre-formulation chapter. We are discussing in the last class about the pre-formulation concepts. Now, as a pharmaceutical technologist, one of the most important job that you are expected to do once you join your workplace as F&D executive is to develop some dosage form. Now, for developing any dosage form, you know very well from your bachelor's degree knowledge that without doing is a significant portion of pre-formulation work, is solid groundwork of pre-formulation studies, you cannot expect to develop a successful solid dosage form. Okay, so the pre-formulation, which is important preliminary work before actually embarking upon the formulation development studies or formulation development practice, we need to do several different types of pre-formulation studies. Now, whether it is an innovator setting, that is the new drug molecule is being formulated into a dosage form, or the generic setting where an existing known drug molecule is being formulated into a dosage form, depending on that, we have discussed in the earlier class also that the quantity and quality of studies will vary to some extent. Of course, in innovator setup, the pre-formulation studies need to be more extensive because we don't know much about the drug or the drug candidate or candidate molecule, and therefore all aspects of pre-formulation need to be done. Now, as we can see from this product development framework, that this pre-formulation in the contemporary pharmaceutics is done in the very initial stage of phase 1, when actually the healthy volunteer work is going on. Healthy volunteer studies are going on. So along with the healthy volunteer, in phase 1, when the healthy volunteers are being given the drug for testing its safety, the pre-formulation work also starts with the hope that in case the drug progresses to the second phase and subsequent phases, then the product will be ready for administration to the patients actually. So during this period, however, keep in mind that the synthesis change. I am assuming that, of course, we are talking about small molecule drug candidates, not biotechnological products. Even with biotechnological products like proteins and peptides also, in the beginning phase, that is during the phase 1, the supply of the drug molecule is very small. Okay, so we need to do a lot of pre-formulation studies. We require a significant quantity of drugs or the drug candidate, but because the synthetic route has not yet been optimized and the production is not running at the full capacity, then naturally the quantity of sample for the API available to the pre-formulation scientist is limited. So for in an innovator setting, when the new drug candidate is being formulated and therefore the pre-formulation needs to be done, the synthetic route has not yet been optimized. Therefore, this quantity of sample available to do the pre-formulation studies is limited. So there is a problem. We need to know all aspects of the drug, but we have very small quantity of formulation available. So in the earlier days, when the advent of advanced analytical techniques were not there, when the scientists did not have access to very sensitive, very sophisticated analytical techniques, then at that time it used to be a very difficult task, and often many important pre-formulation studies need to be skipped due to the lack of sufficient quantity of API. But the situation has changed now. The significant advancement has been achieved in the field of analytical technology, so that even using very small quantity, microgram, nanogram level quantities of drugs, we can go for sophisticated analytical techniques. For example, mass spectroscopy or LCMS mass or other hyphenated techniques are available now to characterize the API, and these systems require very small quantity of the drug. So even in the phase 1 study, when sufficient quantity of drug is not available with the formulation scientists, they still can go for a lot of different types of pre-formulation studies. Now, if we go to the basic pre-formulation cycle that we have seen in the earlier class also, the foremost thing that needs to be done is to go for analytical pre-formulation. Why? Because in the subsequent phases, that is physical, physicochemical, and pharmaceutical phases of pre-formulation, somehow or the other, in every step, we need to analyze the drug. For example, suppose you are going for, testing the dissolution of your product. Now, the drug will come out in the dissolution medium, and you have to analyze the quantity of the drug that has come out. So you need a robust, a validated analytical method for analysis of the compound. Now, before even that, what is need to be done, particularly in case of innovator setting where we are dealing with a new drug or new drug candidate, in that scenario, we are not also knowing about the exact nature of the drug. So we may not be knowing about the actual structure of the drug. We may not be knowing about the elemental composition of the drug. So we need to carry out some extra studies during this analytical pre-formulation stage. For example, here we have the elemental analysis. Okay, here I need to annotate. So like elemental analysis needs to be done to know which elements make up the drug compound or the prospective drug compound. In order to understand the structure of the drug, we need to go for NMR. It may be different types of NMR, hydrogen or PNMR, CNMR, that is to understand the carbon structure. There may be phosphorus present in some molecule, and therefore we need to go for PNMR. And similarly, different types of elements present needs to be understood from the elemental as well as NMR studies, as well as mass spectroscopy. Okay, so the fragmentation of the candidate drug molecule will lead more information, which along with IR value, along with NMR data, along with elemental analysis, will be able to construct the actual structure of the drug. Okay, so all these studies require very small quantity of compounds, but still, if you add it up, it may require overall all the studies may require significant quantity. So in the innovator setting, when the supply of the drug is minimal, these studies becomes quite difficult. So different types of studies such as elemental analysis, NMR spectroscopy, normal UV visible spectroscopy, IR spectroscopy, mass spectroscopy, and the hyphenated techniques associated with mass are required to understand the molecular structure of the compound. Now, depending on the compound, there may be more analytical studies that may need to be done. Now, you see, once these analytical techniques have been established, the molecular structure has been determined, then comes the question of quantification. So here it was identification in the first phase of analytical studies in the innovator setting. Then comes the quantification. So both qualitative analysis as well as quantitative analysis needs to be established, because it is a new molecule. If it is a existing molecule, that is in case of generic setting, these studies need to be done, but that's just for regular documentation purpose. to make sure that the powder or the drug that you are working with is having the same characteristics as the specific standard mentioned in the pharmacopoeia or other official or unofficial standards. So whatever the synthetic chemist has provided about the knowledge about the drug molecule, so whether your product that you are dealing with, your sorry, not product, your API that you are dealing with is having the same structure and other structural molecular aspects are same as that of the standard API. So the next phase is quantification. Quantification means the assay. So you can go for quantification by various means. The most common one is the liquid chromatography, and often this liquid chromatography is combined with mass spectroscopy, that is the LCMS, sometimes hyphenated LCMS-MS. That means the output of the liquid chromatography, the particular pure drug fraction being separated out from impurities is being channeled to mass spectroscopy, and individual fragments of the mass spectroscopy mass spectrometer may be further channelized to another mass. So then it is called as LC-MS-MS, that is hyphenated techniques because there is hyphen in between. So they are in tandem, they are done. So the simplest one may be the TLC. The RF value of the TLC will be useful to identify as well as often quantify using different TLC techniques about the quantity of drug or the assay of the drug. Similarly, if it is a new molecule, then chemical qualitative assays may be developed, some color reaction, etc., to understand the presence of or absence of certain functional groups in it. If it is an existing molecule, these qualitative analysis may be a quick means of identifying as well as finding out the purity of the compound, and quantification is of course ultimately done to declare the purity, how much percentage purity it is. If you remember, I have also told in the earlier classes that now the synthetic routes are so advanced and the purification process is also quite advanced that we do not now actually give more importance to purity. Of course, we know that when we are working with a certain drug molecule, then it is supposed to be pure. Of course, 100%, there is nothing called 100% purity. You should remember that. There must be some kind of impurity which has crept in due to the various routes of synthesis, various solvents being used, various extraction process being used, and none of these are 100% efficient, and therefore some amount of impurity will be there. But in contemporary drug synthesis practice, it is expected that the process is so advanced, the process is so optimized that minuscule quantity of impurity will only be present. But that minuscule quantity may also cause problem in the subsequent phases. Therefore, profiling the impurity of the API is very, very important. It is expected that the API should be as close to 100% pure as possible. But of course, there will be traces of contaminants which also need to be ascertained. Not only that, the drug on storage, whether as API, bulk powder or bulk drug, or in the formulation, will be subjected to different types of stress. It may be thermal stress, temperature variations may be there, some moisture absorption may be there, some light may be incident on the formulation or the bulk drug, and all these will lead to some kind of degradation. So what are the degradants that are formed? That is also that also makes up part of the purity overall purity study. So with time, what are the degradants that are developed? What are the degradation products? How much quantity they are being formed? And of course, a good drug candidate will have minimal quantity of degradants. It should be stable, but unfortunately, not all drugs are having such good property. So in such cases, particularly and in general, in all cases, we need to profile the not only the impurity like the heavy metal or solvent or other solid or liquid impurities, but also the degradation products. Okay, so forced degradation studies are done on the drug if it is a new one particularly, or even if it is a new source. The company of supply company has changed, so it is a new source. Then we need to study the degradation profile profile as well as the impurity profile. So these degradants which are present should be, the impurities that are present should be, because degradants are also impurities, they should be below a certain limit, which should not cause any adverse effect on the integrity of the molecular structure of the pure drug. So all these studies, we need to quantify, okay, how much quantity of degradants are there. We need to identify. For identification, again, all these NMR, elemental analysis, mass spec, IR, EV, etc. are done. For impurity profiling, also for solvent analysis, we have to go for GC, gas chromatography. We have to test for residual water, say Kull-Fischer titration and other things we need to do. We need to go for heavy metal analysis, maybe atomic absorption spectrophotometry or other suitable methods. So we need to quantify in each stage what are the how much quantity of API is present in the supplied sample of the API drug candidate, or as well as the impurities and degradants. So in each of these studies, the HPLC, UV, TLC or similar types of quantitative analytical techniques need to be followed, and therefore even before we can embark upon the purity study, just after the molecular identification is over, we need to develop suitable quantitative methods which are robust as well as validated in order to carry on the subsequent steps such as purity study and other physical, physiochemical and pharmaceutical preformulation studies. So the analytical preformulation is as important as any other part of the preformulation. Often maybe the formulation scientist who is from a pharmaceutics background may not need to always go for these analytical studies. For that purpose, the company might have employed the analytical department. However, in a smaller company, in a small setup, maybe the formulation development scientist who is in charge of reformulation, he or she may need to do it on their own if the company is smaller, and then you have to do everything right from the beginning. So after the analytical preformulation, we are having the knowledge about the identity of the API. We know that yes, this API is the one. Just a minute. Okay, sorry for the interruption. So after the analytical preformulation phase, we know about the identity of the API. In innovator setting, we have found out the new drug structure, its elemental composition. We have found out also the qualitative and quantitative methods of analysis of the drug. We have developed validated and robust analytical techniques, whether it is HPLC or LCMS or other hyphenated techniques. It may be simple TLC also. And we have also known about the purity profile of the drug, which includes both the impurity profile, which again includes the degradation profile of the molecule under different stress condition that it is supposed to, be subjected in the subsequent phases. So therefore, after we have ensured that the API is safe, pure, we have identified the drug correctly, then we move on to the next phase. Okay, so in the initial phase, just a minute. Okay, so in the initial phase, you see these different analytical studies we have done. Okay. Apart from identification, quantification, or assay development and purity study, we also need to do microbiological contamination study. This is very vital in case of particularly the biotechnological products, because the biotechnological products such as the interferons, the proteins, the peptides, the DNA or the RNA, which are used as drugs or API, they are more vulnerable to microbiological contamination. Of course, the natural products, the phytochemicals, if any drug is a phytochemical, then natural products also contains a lot of bioburden. That means bacteria, virus, fungi, etc. may be present due to the natural source of the API. Even with synthetic molecules also, small synthetic molecules also have the same of the problem. Initially during synthesis, there may not be contaminants of microbiological origin, but during the handling, subsequent storage, depending on the nature of the molecule, microbiological growth may happen. For example, if suppose a drug is highly hygroscopic, then naturally with time fungal growth may happen, bacterial growth may happen in such stored samples. So therefore, even microbiological profiling is also vital. This becomes more important in case of natural origin drugs, phytochemicals and other mineral or animal origin drugs, as well as in case of biotechnological products such as the proteins, peptides, DNA, RNA, interferons, etc. In the analytical studies, another point which often is neglected is the organoleptic properties. You know, the organoleptic properties of an API are the properties related to our sense organs, that is the taste, the smell, the texture, the feel. These are some of the organoleptic properties that needs the color. These are some of the organoleptic properties which need to be, studied in the very beginning. Particularly in case of innovator setting, the drug molecule, the drug candidate which is being investigated under the analytical preformulation studies, we need to establish certain standards in order to subsequently carry on the work. So what are those standards? Those standards include all these identification, characterization procedures, and their specifications, quantification procedure, assay development procedure, purity, impurity profile, profiling, microbiological profiling. Of course, apart from that, these physical and organoleptic properties such as the taste, smell, color also need to be, standardized. Okay, because once you have standardized, then you can just by looking, say if we have standardized that the new drug candidate is of yellow color, and then if I am given a white sample, I will immediately know that something is wrong. Suppose the compound is yellow in the pure state, and on degradation its yellowish nature degrades and it becomes whitish in nature, then just by looking at the sample itself we can say that okay, some problem has happened with the compound and it is no longer suitable or fit for formulation into a suitable dosage form. So we have to take again new sample. We have often seen that particularly photosensitive samples are prone to such type of degradation on storage. I myself have some experience with such organic. Oh, just a minute. Sorry for again interruption because people are coming. So I was talking about my experience. There was one drug, amitriptyline, which is if you check the internet, it is highly photosensitive. So we got one sample from a company who supplied us the gift sample, and somehow the person who had packed it may not have taken suitable precautions. Consequently, what happened that the packet, once we opened, it was already degraded. How we came to know? Because the original color had faded and it has become brownish or charred type color. It happened. So organoleptic knowledge about organoleptic properties is very vital. Okay, so often we see that these organoleptic properties like texture, like ibuprofen is supposed to be having crystalline nature. So if you check the powder of the ibuprofen, you will find that it is shiny, small crystals are present. However, what happens is often we find some samples where the powdery nature is more, it is more fluffy and whitish in nature rather than shiny crystalline nature. So we suspect some kind of problem might be there, or some kind of polymorphic changes might have happened, which often is a case. So in the very beginning of the API's journey, during the analytical pre-formulation phase, if we can characterize and standardize these properties, then these becomes very handy tools in the subsequent process, in the subsequent pre-formulation phases, as well as in formulation development studies, as a quick measure of understanding whether everything is okay, going as expected, or some problem has happened. So next one is your physical studies. Okay, so now one thing here, if I go back, there is much less time to deal with each and every aspect of this molecular identification, quantification, purity, microbiological part, or the organoleptic properties. What you need to do is, because questions come in such way that you need to go through the each and every aspects of these studies, that is the analytical studies, physical studies, physiochemical studies, as well as the pharmaceutical studies. We may not have sufficient time to deal with each and every parameter and how they are tested, what are the procedure, what are the techniques. Okay, so it becomes a little difficult often. So therefore, what you need to do is, you need to go through the book very minutely, so that you can have an idea about your the studies that need to be done. For example, what I have already told, that for molecular identification, we need to go for different types of analytical, advanced analytical or instrumental studies. So we know UV visible, you need to have some idea about what is UV visible spectroscopy. We know we use infrared spectroscopy. So now what are the techniques of infrared spectroscopy? In brief, of course, but you need to know. So we need to know that if it is a solid sample, we have to mix the sample with potassium bromide, and then make a pellet, and then go for infrared spectroscopy. Infrared spectroscopy may be simple infrared spectroscopy, or nowadays what we do is Fourier transform infrared spectroscopy, or FTIR. Then there is advanced ATR is there, attenuated total reflectance infrared spectroscopy has come. Okay, so then NMR is there. What are different types of NMR? Whether for what purpose PNMR or proton NMR is done, for what purpose CNMR or carbon NMR is done, for what purpose PNMR or phosphorus NMR is done. So all these in brief you need to know. Okay, what is the basic concept of the fragmentation in mass spectroscopy? So these things you need to go through, because we do not have time to go for each and every aspect in detail in your syllabus. As I have already told, only 10 hours is provided for this pre-formulation chapter, and there is lots and lots of things that needs to be discussed. For example, HPLC. When we talk about HPLC, we need to know what it is, what is its basic operation. You have already studied all these things in your BPharm, but just you need to recollect those and have an idea, so that you can write when it is asked for. What are the different detection techniques? What are the detectors available? Like UV visible detectors are there in case of HPLC. We have the polarimetric detectors. We have refractometric detectors, different types of diode detectors. So different types of detectors. What are the detectors? So these needs to be known. Why? Because depending on the type of the drug, you may need to, suppose you are doing HPLC and the nature of the drug is such that it cannot be, it is not having UV visible spectrum. Okay, so not all drugs will have their UV visible spectrum. You might be knowing that a drug on having some kind of alternative double bonds only have the oxochrome group. That means the color producing group. So whether it is a color or the electronic transition leading to UV absorption will lead to the peak in the UV visible spectrum. But suppose some drug is there which is not having such structural speciality in its structure, in its molecule, then UV visible spectroscopy may not be possible to be used. So in such case, what are the other things that can be used? For example, if the drug is having a chiral center, we may use polarimeter to study the optical property of the drug. Some drugs may be required to be studied by refractometry. For example, if you want to study about honey, for example, you can use a refractometer to analyze its purity and other aspects. So depending on the API that you are working with, or the excipients that you are working with, and what is your goal, so you need to have idea about which API or which chemical or which excipient can be tested by which method. Of course, you will have to go for literature review before embarking upon your actual lab work. But during that time, you need to have an idea, you need to have an observation, background knowledge about the analytical techniques. So for a new API, therefore, these information are usually not available, and consequently, you have to develop it during the analytical preformulation phase. For existing API that is in the generic setting, you already know from literature review what are the different analytical methods available, which one is good, which one is bad, or which one is more accurate, more precise, which one is having more sensitivity, etc., etc. You know, but then this knowledge needs to be translated into your actual laboratory work. So in such case also, sorry, in such case also you need to develop the actual analytical method in your own lab. And you know perhaps that from lab to lab there is a lot of variation. So even though a validated method is available, SOP is available for a particular API with a particular analytical technique, say HPLC, you need to develop and validate your own system. In your own system, maybe the method will be same, but you have to validate it once you transfer it to your own system in your own HPLC machine. Even if the machine is changed, suppose a machine breaks down, another new machine comes. So in that case also you have to again revalidate the analytical procedures. Okay, so these things need to be kept in mind. So being pharmaceutic students, you cannot just close your eyes to analytical chemistry. And that's why in the very first class of your MPharm, I have told you that as pharmaceutics majors, you need to have idea about most of the other pharmacy fields. Yes, if anybody is having some question. Is anybody having some question? You can raise hand if you want to ask something. Or you can unmute yourself and ask, there is no issue. Well, teaching, if you have, yes, tell me.
Sir, exam এ একটা question ছিল যে pre-formulation technique-টা কোন trial-এ use করা হচ্ছে, মানে
কোন trial-এ মানে কোন phase-এ?
হ্যাঁ, কোন phase-এ, sorry,
খুবই ইজি। এই যে আমি যেটা বলেছিলাম, এই যে এখানে যে ছবিটা ছিল সেই ছবিতেই তো দেয়া রয়েছে pre-formulation-টা কোন
ছবিতে দেওয়া আছে phase 1-এ, phase 1-ই করে এসেছি।
রাইট, কারেক্ট। আনসার তো পেয়ে গেছিস, আনসার দেখিসনি? হ্যালো, হ্যাঁ বলছি আনসার পেয়েছিস না? আনসার পাঠানো হয়নি?
কই না তো।
আচ্ছা, তাহলে আমি আজকে রিলিজ করে দেবো, আনসার গুলো রিলিজ করে দিলে তোরা দেখতে পাবি, আমি রিলিজ করে দেবো ওখানে। তো so pre-formulation, actually there is no fixed phase. Okay, we start in phase 1, but as we go through different phases, there will be multiple times we may need to go for the pre-formulation. কারণ what happens is as we progress with the studies, as we develop more information about the API, particularly in the innovator setting, we may find out that certain characteristics of the molecules are prohibitive or not prohibitive, maybe creating hurdle in developing a suitable or successful dosage form. For example, most often what happens is the type of salt that you choose to make and formulate may be having some issues. Okay, for example, if you suppose choose a sulfate salt, it might be having hygroscopicity issue. But if you choose a hydrochloride salt, it might not be having the issue. If the drug is having some solubility problem, then choosing a suitable salt may solve the problem. So once you choose the particular salt, you change over from, say, sulfate salt to your hydrochloride salt, or sulfate salt to mesylamine salt, or sulfate salt to some other salt, acetate salt or some other salt, whatever, depending on the particular drug molecule, you will see that some of the characteristics, the physical as well as physiochemical characteristics changes. The solubility changes, the polymorphic nature may change, the crystal characteristics may change. So you need to again redo some phases of your pre-formulation. So we cannot exactly say that it is in the phase 1 only where it will stop. A good company is one who continuously keeps a tab on the progress of the drug molecule, learns from the data that is generated, learns from the market. Even after phase 4, they learn from the market issues, okay, and then they will try to update it. Of course, the more intensive studies that has been done in this phase, pre-formulation, this phase, the more the less problem it is expected to come in the subsequent phases. Okay.
Sir, নেটে এটা প্রিক্লিনিক্যাল আর ফেজ 1 দুটোই দেখাচ্ছিল।
Yes, প্রিক্লিনিক্যাল আমি এটা বলেছিলাম, এটা তো জাস্ট clinical phase টা দেখানো আছে. What is preclinical? Preclinical, there are again 2 phases. Okay, there are 2 phases. One is 00 phase, one is second one is single zero phase. Okay, 00 and single zero. Now in the 00 phase, we actually go for in vitro studies, biochemical studies, and single zero phase usually we go for animal studies. So during that also the company may try to develop the pre-formulation characteristics. But here again there is something, not always it is done. Why? Because sometimes the drug molecule may be the innovator company may not be the innovator. Maybe some university department has first worked with the pre-formulation work of the not pre-formulation, preclinical part of the work of the API. So in that case they might not be interested they might not be interested to know about the pre-formulation. Suppose a synthetic chemistry lab has stumbled upon a new API. Okay, now they won't be interested in doing the pre-formulation. They'll be more interested in the identification, characterization, analytical development of the molecule. So after a certain level of work has been done, maybe they will be collaborating with a pharmacology department to know the animal study, what is the result of the animal study. So therefore pre-formulation, preclinical phases, pre-formulation is required if it is in the will be done if it is in the company setting. But again, during pre-formulation, in the very early phase, preclinical as well as phase 1, as I told some time back, that in an innovator setting, the new molecule is very scarce in nature. Very little quantity, maybe milligram level quantities are available. So you may not be always be able to do all the studies that you want to do. So therefore, whether it is preclinical or phase 1, it depends on the availability of the quantity of drug that will dictate how much pre-formulation will be done in preclinical phase and how much in phase 1. Of course, keep in mind, it's just a business interest that when you are in preclinical 00 or single zero phase, you are not sure whether you will get the NDA. Okay, whether you will get the approval to your NDA, that is new drug application. So if you do not get the NDA, there is no point in going for further cost incurring by doing the pre-formulation study. Only when the NDA is approved, then only when it enters into the phase 1, pre-formulation becomes more meaningful, because until and unless we can get the NDA approved, we cannot put it into humans in the end. So naturally the pre-formulation will have not much value. Of course, when you can argue that why not give it to the animals? Yes, we can give, but most of the time, depending on the route of administration, a simple solution, suspension, etc. is enough to know the effect of the drug on the animal. Okay, so only when it comes to the humans that we need to also consider the proper dosage form which has to be administered to the patient. We cannot simply put anything, all drugs in solution or suspension form, because of patient compliance reason, adherence to regimen reason, and other reasons are there. So therefore, the serious, or I can say more planned and active pre-formulation study will be carried out during the phase 1 primarily. Okay, but there is no harm, depending on the company's policy, company's economic strength, and the future that is expected of the drug. Someone can start in phase preclinical phases also. Okay, but mostly in phase 1, pre-formulation will start, because at that time it becomes more meaningful, at least from an economic point of view. Without knowing whether the drug can be really administered to the patient, without getting the NDA approved, new drug application approved, there is not much point always in doing the pre-formulation, because if it fails during the preclinical phase only, there is no way that we need to develop. There is no point in developing a new dosage form, because we cannot give it to the patient. Am I clear? Is your query satisfied?
Yes, sir.
Okay. Okay. So moving on. So these analytical studies we were talking about, these analytical studies, you need to go through at least a customary loop with little bit of information on each. I will tell rather read the books as well as make your own scribbling notes, because each one of you might be knowing something about some analysis and nothing about some other analysis. So depending on your strength and weakness, you may know UV visible very well, but you may not know HPLC very well. You may know HPLC well, but you may not know HPTLC very well. So depending on where you are having the limitation, you do your own notes, because there is no end to it. You cannot actually, if you want to write a note for everything, then of course it will become volumes of books. So what I suggest is, depending on your weakness and strength, you make your own scribbling notes, scribble something while reading the books, so that you know some of the things you already have the knowledge, and the things where you do not have the knowledge, you put it down in writing for at least the exam purpose. Okay, so these things are vital, that depending on your background knowledge, you decide how much in depth you will need to study. It's very difficult, because I cannot do justice to the whole syllabus if I want to go very deep in each aspect. It can be done, but then it requires several months. Similarly, same thing about not only identification and characterization, but also about the purity. Of course, as I already told you, until and unless the analytical technique, valid, robust analytical technique has been developed, we cannot go into the purity phase. Okay, so and also other requirements, such as the microbiological contamination requirement, requires some more microbiological studies, which is entirely different from the analytical chemistry studies, and of course requires the support from the microbiology department. For taste, smell, texture, that is the organoleptic properties, also you have now specialized equipment, such as the electronic nose for quantification of at least, not if not quantification, but at least for maintaining uniformity of taste, smell. Electronic tongue is there for quantification and equal distribution of not distribution for quantification, as well as uniformity from batch to batch about the taste of the sample. Like that, if a lot of different things may be required to be done depending on the particular drug. Now next one that is important is your physical studies. Now again here, physical studies, the primary physical study that is done during the pre-formulation phase is of course solid state property, but keep in mind this is given more importance because of one very simple reason, that 95% or more, or even I can say 99% of the dosage forms that are sold in the market are primarily solids. So we call about solid state properties. However, there are other physical states of the dosage form, such as the liquids are there, aerosols are there. So depending on the intended final dosage form that the company is having a plan to market, the physical studies will also vary. Okay. However, because most of the drugs are solid except a few, we need to go for the solid state characterization in any case, whether the drug will be ultimately given as tablet or will be given as an elixir or a solution or a suspension or an aerosol, whatever may be, because mostly most of the drugs that are available, the APIs that are available are in solid state primarily. The raw material is solid state, so we need to go for solid state properties. Now there are several solid state properties. Let us go back to this figure again. You can see from here about the different physical characterization. Of course, this is not exhaustive. I told you in the very beginning, the different physical characteristics that need to be studied, analyzed. So one of the most cheapest, the easiest study in physical analysis, but very vital study, often people forget to do in the academic setup, but very vital, is the melting point determination. Okay. Now it is solid, that's why melting point determination. If the drug was a liquid, then we need to go for boiling point determination, and accordingly you have to choose. So because most of the drugs that you will encounter is solid, so you need to know melting point, how the melting point is done, like whether you will do it in Thiel's tube or you will do it in a analytical quality automatic melting point apparatus or a visual melting point apparatus. So different types of apparatus you need to know. Then you need to know what constitutes a accurate melting point. Usually it is known to you perhaps that usually we say that the melting point of the drug is say X plus minus 2 degrees centigrade. Okay, so usually plus minus 1 degree centigrade. So usually if the melting point of a drug is say 172 degrees centigrade, then naturally we say that the drug is almost pure if the melting point comes between 171 to 173 degree centigrade. You know, when you have done melting point study, in the beginning there is a starting point when the material starts melting, and there is a end point where the material has completely melted. So the difference between these two values, the starting temperature and the end temperature of melting, should not be more than 2 degrees centigrade in the ideal case. However, depending on the quantity of impurity that are present, quantity of degradation products that have already developed in these sample, the melting point may become a little bit more. Another very important aspects where the melting point changes, please keep in mind, is when the during storage, the drug's solid state form changes its polymorphic state. Okay, so you know polymorphism, you know the existence of a chemical compound in different physical forms. Some may be crystalline, some may be amorphous, some may be solvates, some may be hydrates. Different forms are available. So melting point significantly changes if during storage the polymorphic status has changed. You have already studied in the form that usually the metastable form of any polymorph is more highly soluble, whereas the stable form is less soluble and more inert. and less prone to changes. So you may be tempted to use the stable form. It may, in some cases, be perfectly meaningful to go for the stable polymorphic form, but then metastable form is having higher solubility because it is less stable, so the molecules quickly go into solution and move away from their own crystal habitat. So therefore, most of the time, in order to maintain the proper bioavailability, which is again dependent on the solubility of the drug from the dosage form into the biological media. So we usually take we usually take the metastable form. Just a minute. Okay, again, sorry for the interruption. So what happens is, so these polymorphic changes have to be kept in mind and has to be monitored. And melting point is a very simple and straightforward and maybe a quick method also to check the polymorphic nature of the drug, as well as to check the changes in the polymorphic nature of the drug on standing, at least as long as it is available as a raw material, as a powder. Okay, so how to do the melting point, what are the apparatus that are available, what are the precautions that need to be taken during the melting point study, all these things need to be understood, know, and because obviously if you are going into an FND, you will need to do this study. Okay. The other important parameters that you should go for are the characterization of the polymorph of IR through IR spectroscopy, Raman spectroscopy. Along with the vibrational spectroscopies, you have to go for differential scanning calorimetry, X-ray diffraction study. So usually what we do is, when we work in our, say, even inform projects also, to understand the crystal nature and polymorphic nature of the drug, we take the data from melting point, we take the data from IR. Raman we may not always get, but if you get, it is better. We take the data from DSC, we take the data from XRD, and all the data we combine together and build the picture or draw the picture of the crystal form or the polymorphic nature of the drug. Often this polymorphic nature is critical in deciding the success of the formulation. It may make or it may break the formulation stability and other stability, and most importantly efficacy also, if the polymorphic nature changes. Why? Because the different polymorphs of a drug, although having the same chemical structure as per definition, vary significantly in terms of solubility and therefore dissolution and hence bioavailability. Okay, you might have learned about the different dissolution methods in your BFAM, and they are the different dissolution. In the dissolution theories, you might have probably learned about how the dissolution, why the dissolution varies from one polymorph to the other. Okay, I'm not going into the details. It's an interesting fact, but you might have already studied in BFAM. So these different techniques, such as the DSC, XRD, need to be regularly done. Okay, we need to regularly do such techniques. Apart from that, we also do several other studies. For example, partition coefficient we do. Okay, so we go for log P studies. Log P studies means the KOW, that is partition coefficient between oil and water. Often our body does not have pure water. We have some kind of buffer. We have saline solution in our body, right? The body contains a lot of salt. So basically, log P may not be a proper indicator always. So we go for log D, that is distribution coefficient, which is basically log P with water being replaced by isotonic phosphate buffer saline, which represents the actual biological fluid that is present in our body. So the partition coefficient is also an important aspect. In the innovator setting, where you have very scant knowledge of your drug molecule, you may not also or even know what is the solubility of the drug. So you have to study about the solubility of the drug, and keep in mind, solubility study is not a simple study, although the things appears to be simple. There are methods to test the solubility, which may extend up to several hours, several days, several weeks, because as the solubility of a drug falls, the time for solubilization increases. We need to give more time to accurately determine how much of the drug will be soluble in a particular solvent. And different solvent solubility may need to be studied. Along with that solubility, we may also study the intrinsic dissolution of the drug. That will become vital in deciding the excipients that will be required to be put together with the drug in order to make a tablet or similar other solid dosage forms. Apart from these crystalline investigations like the polymorphic or amorphous nature identification and quantification, we also have another thing, that is salt selection. Okay, the salt form that is required to be, used, that as I told just some time back, is also becomes vital. Not only is the change in salt form leads to different crystalline nature, that is polymorphic changes, different crystalline characteristics, it also has a very significant effect on the solubility and hence the bioavailability. So one salt may be having more bioavailability, another may be having less bioavailability. Another thing is, as I told, the stability, the hygroscopicity, the moisture absorbing capacity of the drug may change with the selection of the salt form. So a judicious selection of the salt form is vital. Another very important aspect with salt form selection is the effect on the crystalline nature and the effect of crystalline nature on the compressibility of the powder into tablet. Okay, so usually we might be studying already in another part of your physical pharmaceutics, modern pharmaceutics syllabus, that this crystalline powders can be compressed well into formed tablets, whereas amorphous powders are less compressible, and therefore it becomes difficult to compress the dose into a suitable sized stable tablet. Okay, for example, your very common drug paracetamol, we take very often when we have fever, but many of us may not be knowing that paracetamol is quite very difficult to compress, because it's very fluffy and amorphous in nature, and its crystalline structure is very weak, and therefore what happens is it's very difficult to compress. So it's we can call it an incompressible powder, and therefore we require extra excipients such as the MCC, microcrystalline cellulose, or direct compressible starch, etc., to successfully form a suitable tablet from such incompressible or poorly compressible powders such as paracetamol. Okay, so these information will be available when during the pre-formulation phase, and this information will help us in developing the proper final dosage form. Okay, so of course, if we want to go for, say, liquid dosage form, and we definitely will want to take a salt, use a salt which will be stable in the liquid formulation and in the solvents that are being used. And we know that in case of oral formulation or even parenteral also, we have to use some salt which is soluble, because our body is water made up of 70% water. We cannot use a salt which is insoluble in our biological system or in aqueous fluids. So therefore salt selection is quite important, and it's a tedious job to finally find out which salt is having all the desired properties, such as the solubility, the nature of hygroscopicity, the propensity to undergo degradation, hydrolysis, etc., oxidation, etc. So a salt which is having the least of these and maximum of solubility will be logically selected, if not other aspects are there. Not only these, but also the compressibility, in case you want to make a tablet, becomes a vital factor in case of your salt form selection. So you should not neglect the salt form selection also. And how you will find out the salt form? Apart from the analytical tests such as the qualitative say salt test or quantitative assay with suitable analytical technique like HPLC, TLC, etc. You also have the melting point as a very important tool to know which salt it is. Because of course that is in generic setting, or you have already developed the salt and then testing the melting point. So in generic setting, just by doing certain melting point study, you can often say which salt it is, if you know that generally there is significant difference between the melting points of different salts of the same compound. Okay. So we have discussed about the salt form, we have discussed about the crystalline state. Okay, in this polymorphism aspects, the last 3 points, as you can last 2 points, as you can see, the high throughput screening and computational screening. This in brief I have already discussed in the last class. So it is a generalized thing that using the advanced computers and the high throughput systems, we can go for in silico screening of the various polymorphs that we need to develop, and may be possible first of all, and then we need to develop. And through HTS we can check a lot of different polymorphs in small quantities for each, and finding out which one is having the desired properties in very short period of time. Earlier, by the usual pre-formulation route, we used to take a lot of time to study each and every polymorph that is that makes up the particular API. But now using computational screening as well as high throughput screening, we can very quickly, if not very quickly, but we can quickly, compared to previous generation, we can quickly say which one is suitable for progression to the next phase, to the advanced pre-formulation and then to formulation design. So, so apart from this, as I already told, the hygroscopicity is important. Hygroscopicity changes a lot of different properties of the drug. One is, of course, moisture absorption may lead to degradation. Okay. Second is the hygroscopicity often changes the tableting behavior. So if it is a solid dosage form, we need to develop a tablet or a capsule. In that case, the hygroscopicity plays a significant role. A hygroscopic material will be having solubility issues also, because already it is having some water absorbed into it. So it might be having issues with solubility as well. Okay, so therefore what we need to do is we need to understand the fact that hygroscopicity, which is quite simply measured using a moisture balance, which you have seen in our laboratory also, becomes an important part. The stability, overall stability of the API as a raw bulk material, raw material, as well as in presence of other excipients in the dosage form, may be jeopardized if a molecule is hygroscopic. So therefore what I suggest is, if you suspect that in your laboratory setup, in industry setup, of course you have to do hygroscopicity, but in your laboratory setup, when you are doing your project, if you suspect that there is hygroscopicity issue, then you should always check the hygroscopic potential. Okay, so knowing the hygroscopicity is quite crucial in such cases. Again, I am stressing one point. There is no fixed, procedure or SOP. I cannot say there is any SOP for pre-formulation studies. You have to do it according to the need of the molecule that you are dealing with, the API, as well as the intended excipients that you want to utilize. Okay, so this is vital that you understand the fact that not always all studies that we are teaching here or you study in the books will be required for all molecules, but a subset of these molecules will be required. A drug which we know is very much hydrophobic, naturally we'll have no issues with the hygroscopicity. Until and unless the pharmacopoeia tells you to declare what is the moisture absorption rate, you don't need to, you may not need to go for hygroscopicity study. But as a precaution, because water is a detrimental factor, hydrolysis is a known degradation route for several drugs. So as a precaution, this study should be ideally done if you have the quantity of sample available as well as the availability of infrastructure. The next point which is there is particle characterization. Now, these particle studies, the particle size is very vital. Okay, particle size is very vital. You know, particle size can make or break a particular formulation. Okay, particle size, you know, depends on the particle size will determine the specific surface area or the surface area available for dissolution, and therefore the solubility will be related to the particle size. Okay, so you have already known that. So the more finer a particle, the larger surface area will be available, and the higher solubility you can expect. Okay, so therefore the particle size becomes an important part in determining whether the API will be good for dissolution or not. Okay. Now, what happens is particle size not only determines the dissolution characteristics of the drug, but the size and shape of the particles are vital in other aspects also. Many production aspects may be hampered due to non-optimum particle characteristics. Okay. এক মিনিট, জাস্ট এ মিনিট, আমি এখন একটা ক্লাস নিচ্ছি। Sorry for the introduction again. My ক্লাস সেখান থেকে নেওয়ার থেকে বাড়ি থেকে নেওয়া বেশি সিম্পল হয়, যাই হোক কিছু করার নেই। তো যেটা বলছিলাম যে this particle size not only is important for ensuring proper efficacy by ensuring dissolution and solubility of the drug, but also the size and shape becomes vital in the subsequent production purpose. You all perhaps noted, have studied about the issues of particle shape, particle size being different, which leads to different types of problems. Like if suppose we want to make a tablet and we have the different particle size of the API and the excipients, what will happen? During mixing at different stages, there will be stratification. That means there will be size separation. The bigger particles will fall down to the bottom, and the larger, smaller particles will be at the top, and that will lead to non-uniform distribution, which leads to weight non-uniformity, content non-uniformity, and other issues. Then the shape, irregular shape particles, mostly pharmaceutical particles are irregular shape. So irregular shape particles will have issues of flow. Now we all know that in order to develop a suitable tablet, the uniform size particles should flow uniformly into the from the hopper to the tablet die cavity. Now, if the particle size is not having suitable uniformity, then and shape is also not proper, then there will be locking of particles during the flow. Irregular shaped particles will get locked to each other, and what will happen? There will be the flow being obstructed. So you might have heard about, just go for the whiteboard. You must heard about two terms, that is your arching and rat holing. Have you heard of about these terms during your study of tablets? Arching and rat holing, students?
Yes, sir.
Yes na no, শুনতে পেলাম না।
Yes, sir. Rattling, I've heard.
Okay, so you are কি বললি তুই? Shubhra?
No, sir. Payal.
Payal, good. Anybody else?
Yes, sir.
কতজন জানো rat holing, arching এগুলো পড়েছো?
হ্যাঁ স্যার।
দুজন শুনছি। আচ্ছা, আমাকে বলতো rat holing কাকে বলে, arching কাকে বলে, কারণ এগুলোই ইম্পর্টেন্ট জানা। আমাকে একটু বল, একটু আইডিয়া দে।
স্যার হপারে যখন পাউডার আর কি মানে পাউডার পার্টিকেলস যখন পড়ছে, তারপরে মানে একটা মানে হোল হিসাবে মানে ক্রিয়েট হয়ে যায় ওখানে মাঝখানে পাউডার পার্টিকেলসে।
আর
স্যার
arching?
অ্যাকচুয়ালি হোলের চারপাশে পাউডার পার্টিকেলস গুলো জমা হয়ে যায় এবং একটা অন্য শেপের হোল ক্রিয়েট হয়।
মানে ব্যাপারটা হচ্ছে যে একটা ইয়ে হয়ে যায়, কি বলে, যেটাকে বলে একটা টানেল মতন হয়ে যায়। এটা যদি হপার হয়, এটা যদি হপার হয়, এই যদি হপার হয়, সরি, এইরকম ভাবে দুপাশ দিয়ে, আমার ড্রয়িং ভালো হচ্ছে না, যাই হোক, এদিকে পাউডার, এদিকে পাউডার, আর মাঝখানে এই জায়গাটা হচ্ছে ফ্রি স্পেস হয়ে যায়। কেন না, ইরেগুলার শেপ পার্টিকেলস গুলো এরকম, একটা পার্টিকেল এরকম যদি হয়, আরেকটা পার্টিকেল যদি এরকম হয়, তারা লক হয়ে যায়। ওকে, এটাকে বলে র্যাট হোলিং। আর আরেকটা জিনিস কি, আর্চিং। আর্চিং এ কি, আর্চিং এ এই যে হপার, এখানে মাথায় এরকম আটকে যায়, এদিকে পাউডার, এখানে আটকে যায়, তার মানে কি ও আর ফ্লো করবে না, তাই তো? সো দ্যাট ইজ আর্চিং। মানে যেরকম ব্রিজে দেখেছিস না, ব্রিজ গুলো থাকে কিরকম, এরকম, হাওড়া ব্রিজ দেখেছিস তো, এরকম থাকে তো। তো তো সেটার জন্য বলে আর্চিং, আর্চ। ওকে, তো এগুলো সবই হচ্ছে ইরেগুলার শেপ পার্টিকেলস এর জন্য হয়, এবং ইরেগুলার শেপ পার্টিকেলস গুলো ফ্লো এর সময় একটা আরেকটার সঙ্গে ক্রেভিসেস গুলো লক হয়ে যায়, and because of that locking what happens is the rattling, arching, this type of problem comes. So therefore when you are developing your dosage form, you have to keep in mind that your particles, not only API particles but also the excipient particles are of similar shape and size. Size different hole to hobei stratification বা separation, size separation হয়ে যাবে, কিন্তু shape different হলেও কিন্তু এই প্রবলেম গুলো আসবে। So you have studied about these particle shape and size during your micromenetic study. Have you studied about micromenetics? নামটা মনে পড়ছে?
Yes, sir.
Yes, sir. পড়েছি micromeritics.
Micromeritics is in solid dosage form development, that is very vital, very important. Okay, the particle size distribution, what type of distribution is suitable for what type of particle, brittle particle এর জন্য কি রকম distribution হবে, fibrous particle এর জন্য কি রকম distribution হবে, soft particle এর জন্য কি রকম distribution হবে, এগুলো কিন্তু micromeritics এ পড়েছিস। তারপরে sieve analysis করার পরে how to convert the number average to weight average, Hatch-Quaid equation, এগুলো micromeritics এ আশা করি পড়েছিস। So these are becomes very important when you actually start developing your own product. Okay, so maybe when you have studied you might not have guessed the importance of these basic theory, but when you actually start working, the better you know these theories, the better equipped you will be to solve everyday problem, because in product development every day there will be some problem. Okay, so therefore these basic things which you have studied in the BPharm you should not forget. Problem is I have not much time within your syllabus specification to go deeper and deeper into each and of every aspects of these problem solutions and the methodologies. So you must give a customary reading of whatever you have studied in BPharm related to these pre-formulation, particle size, etc., etc. Okay, so what you need to know also is what are the different methods of analysis of particle size. Now you see, if you want to, I will ask a question. If you want to develop a tablet, what will be the most common particle sizing equipment that you can use? Practical angle theke. From practical angle, what will be the simplest equipment that you should use to know the particle size? You are developing a tablet. Anyone?
Sir, microscope.
Okay, microscopy, yes, you can do that, but I have added a word from practical point of view. Okay, microscope you can definitely do your work in lab scale, we do use that, but what will be the simplest, the quickest? Practical angle মানে simplest, quickest and accurate, দু তিনটেই থাকতে হবে, তাহলে আরেকটা কি হবে?
Sir, it is in powder form.
Obviously you are making a tablet, so it must be in powder
Sorry,
form.
sir, MCC
কোয়েশ্চেনটা
microcrystalline
শুনতে
cellulose.
পেলাম না, একবার বলবেন, নেটওয়ার্ক প্রবলেম।
আচ্ছা বলছি, microcrystalline cellulose তো we are adding to make
Yes,
it
sir.
flow better or
Yes,
compress
sir,
it
yes, sir.
better,
Yes,
but
sir,
my
flow
question is
properties.
flow properties and compressibility, but my question is little different. My question is, you are given the task of developing a tablet, obviously that means you are working with a powder API. Okay, now what type of microscopic technique you are supposed to use to have a practical knowledge about the particle size of your drug and the excipients? Questionটা শোনা গেল এবারে?
ইলেকট্রন মাইক্রোস্কোপি, অপটিক্যাল মাইক্রোস্কোপি।
Electron microscopy, optical microscopy. Okay, anybody else? Anybody else? Just tell what is in your heart, what you know, just tell
Sir,
it.
it's scanning electron microscope.
Scanning electron microscopy. Okay,
So
I'm happy that
other
yeah.
than microscopy, sieving method is there, sedimentation.
Exactly,
Yes, sir.
sieving. Yes,
Sieving method.
all these techniques, I am happy to know that you know these techniques, that is very good, but you have to also keep in mind that economy and speed is an issue. You cannot give the sample to scanning electron microscope department. Your company might not be having it. You have to give it to some other company, and they will take some time. So the simplest, the quickest, and for your purpose accurate also to some extent is the simple sieving. You will sieve it and you will find it. Okay, but same question if I put not for tablet but for say a suspension or an aerosol, then what will be your answer? Obviously you can understand from my question that it is not sieving anymore. So what will be the next answer?
So sedimentation.
Sedimentation, for particularly for which dosage form?
Suspension.
Suspension. Good. Anything else?
Sir, for emulsion, we use microscope.
Okay, for laboratory purpose it is fine, but there are certain emulsions where particle size, droplet size is so small that you might not be able to use the optical microscope to view it. It might be so small. Okay, so I have already given a hint. So anybody else can name
Scanning
another technique?
electron microscope.
Scanning electron microscope may be used, but there are issues with scanning electron microscope. For example, if you want to use say a emulsion, study emulsion droplet size, then same is not a possibility, because liquid cannot be studied through scanning electron microscopy. You have to go for fridge fracture scanning electron microscopy. Okay, FF same. Again, it depending on the type of the sample that you have, it may or may not be possible. What else is there? Any guess?
Filtration.
Filtration করে কি করে মাপবি? আইডিয়াটা বল।
সাসপেনশনের পার্টিকেল গুলোকে ফিল্ট্রেশন করে।
সাসপেনশনের পার্টিকেল গুলোকে ফিল্ট্রেশন করে করতে পারিস, বাট ফিল্টার করলি দিয়ে তারপরে পার্টিকেল গুলোকে তো আবার ফিজিক্যালি মাপতে হবে। কারণ ধর তুই বললি আমার পার্টিকেল সাইজ 0.45 মাইক্রনের থেকে বড়, তাহলে তুই 0.45 মাইক্রনের একটা ফিল্টার দিয়ে you will be filtering and you will be showing, see my particles have been filtered through 0.45 micron, so my particle size is 0.45. But can we tell that? Not quite, because some will be bigger than 0.45, but how much bigger? Whether it is 1 micron, 2 micron, 5 micron, we do not know. We know that it is bigger than 0.45 micron.
Yes, sir.
Right? Like that, we can use several filter paper of different porosity, but that is not a very easy process. Okay, you have to filter through different filter papers, and then you have to weigh each filter paper, and then find out how much sediment has been deposited, and then it may not be. It is the combined particle size of the drug, API, etc. all together, but not individual. So what you can do?
Yes,
So
sir.
there is one method, Coulter counter method, by which we measure the size and volume of
Yes,
the
very
particle.
good, very good, very good. Coulter counter method. What is the technique? When a basic principle of Coulter counter?
So ওখানে একটা নিচে মানে
No, I'm not talking about the description of the instrument. What is the basic principle of Coulter counter?
conductivity
Conductivity, yes, changes in conductivity. আমি যেগুলো জিজ্ঞেস করছি ইন্টারভিউতে কিন্তু এরকম ছোট কোশ্চেনই জিজ্ঞেস করবে, and you have to be very careful about what you are answering. এই যে এখনি তুই মেথডোলজির বদলে প্রিন্সিপালের বদলে মেথডোলজি বলছিস, এইগুলো কিন্তু খেয়াল রাখতে হবে যে কি জিজ্ঞেস করছে আমি সেটা বলবো, নাহলে ইন্টারভিউয়ার কি চিন্তা করে যে ও বোধহয় জানে না, ঘোরানোর চেষ্টা করছে। সো বি টু দ্য পয়েন্ট। ওকে, আমি দুটো বাজলে এগুলো জানার জিনিস। ওকে, সো দিস why I am discussing these, because with not only particle size but also with all the different analytical pre-formulation studies that you are going to need, you need to know about what are the techniques, what are the methodology in each technique, what is the principle, what is the instrument used, for what type of sample you need to go for, what type of analytical method. These things are important, and as you can understand, within 10 hours I cannot teach you everything, right? And there is no need also as per the syllabus, because in B.Pharm you have studied it. So you need to study these as well, even in M.Pharm also. If you know, very well. If you don't know, again you have to go through. Okay. Now coming back to my question, that is for tablet. Okay, we have used microscopy. This CV. But for aerosol or a suspension, we may go for Anderson pipette, that is sedimentation rate, but that is again a slow process. So nowadays what is done is, with advent of technology, we now use this technique called DLS. Okay, DLS or QLS also we say, QELS we say, QELS, DLS or QLS. Diffuse light spectroscopy or quasi-elastic light scattering, diffuse light scattering or quasi-electric light scattering. It's a kind of spectroscopy where laser light is incident on the particles that are suspended in a suitable solvent, and they are reflected on refracted and transmitted to different detectors, and from there, using a technique called Mai's theory. Okay, Mai's theory, we go for analysis of the particle size. It's a very quick and very accurate method of analysis of particle size, and is useful, very much useful for all types of liquid dosage forms, to be very specific. Also, those companies who can afford, it's a quite costly instrument, minimum cost is more than 25 lakhs. So those companies which can afford, for them, even they have now replaced this microscopy or sieve analysis by this method. The machine is simple, like a UV visible spectrophotometer. It has a cuvette, just like UV visible spectrophotometer. You put your sample suspended in a liquid in there, and then you put the cuvette containing the sample into the machine, put on the machine, let it run for a certain cycles, maybe 100 scans, etc., which is very quick, and then it will give you the result using suitable graphical as well as tabular form. From there, you can know what is the particle size, what is the distribution, what is the polydispersity index, that is how much equalness of particles is there in the sample, etc., etc., and in quite quick way. So nowadays, this QELS or DLS principle, quasi-elastic light scattering, or DLS principle, is used. And the machine name often is the laser light scattering machine, or laser light diffraction machine, or we call it by their brand names like Malvern particle size analyzer, or Horiba particle size analyzer, like that. Metro particle size analyzer, like that we call it. So this machine is a very advanced machine. Now there are again certain problems, like suppose you have a gel and you have suspended particles in the gel, or you have some droplets, nano gel, so some nano emulsion has been jellified. Now if you want to analyze using this DLS diffuse reflective light scattering technique, what will happen is you have to dilute the gel. And as soon as you dilute the gel, the particle size, the droplet size changes. So it's the original size will not be available. So for such purpose, there is ultrasonic particle size analyzers have also come nowadays, which instead of doing any kind of sample preparation, you just simply use ultrasonic wave passing through the gel. These are again useful for gel, paste, ointment, etc. Solid or semi-solid, semi-solid dosage forms particularly, even can be done for liquid also, but primarily done for semi-solid. So you can, without diluting the sample, you can simply use ultrasonic spectroscopy to find out the particle size and distribution. It's something like the ultrasonography that we do when we are ill. Okay, so these advanced techniques, right from microscopy to sieving to Anderson pipette, that is sedimentation technique, to Coulter counter, that is conductivity method, to laser light scattering or quasi-elastic light scattering technique, to this ultrasonic technique, you should know all the techniques, at least names and the basics of each technique, the basic principle in brief, as well as the instruments that are used. Okay, so like if I talk about conductivity method of measurement of particle size, you will immediately tell Coulter counter. If I talk about the optical method, you will immediately say optical microscopy. If I talk about electrical resistance, again you will talk about Coulter counter. If you talk about light scattering, you will immediately talk about the laser light scattering spectroscopy. So these things are vital and you should know. Okay, so of course I can go on for quite a long time. There is not much time. We are around 10 minutes to 12. We'll end here today, but keep in mind that whatever methods that we are discussing, we are just giving the basic idea. It is the tip of the iceberg, but you need to study about the individual methods in brief. Their basic methodology, their basic principle. You should know about the instruments that are used and the applications for which they are primarily used, if not all. যে যত বেশি পড়তে পারবে ভালো, কিন্তু প্রচুর আছে বলে আমি বলছি মিনিমাম এই জিনিসগুলো যেন তোমরা শিখে রাখো, কারণ ক্লাসে সবকটা নিয়ে ডিসকাস করার মতন সিলেবাস টাইম দেয়নি, because the syllabus is expecting that you have already studied this in your bachelor's degree. So we will go into the next generation or next level of the knowledge that is expected from a master's student. Okay, so I'll now take on questions if any more is there, and I'll not go forward with the topic anymore. So we have talked about the physical aspects to some extent. There are again some more is there, because of paucity of time we'll not go into that. So anybody having question, you please ask. কারো কোনো কোশ্চেন থাকলে জিজ্ঞেস করতে পারিস আজকে যা পড়ানো হলো। No question. If there is no question, in that case I'll stop the meeting. Okay, fine then. So see you again next week. I think I have to one day contact with you to classes. So go through these books. I don't recommend you to just go for studying. I will recommend you to just go through these chapters just like storybook, because there are huge quantity of information that needs to be absorbed, comprehended. So don't go for with the thinking of that I have to know this. Just like a storybook, go through the material, you will see, you will retain it and be happy and enjoy what you are studying. You will see, apart from what you have studied in B.Pharm, a lot of other things are there. I have given the links for the important books that is required. Some more will be given as time goes on and you move into the syllabus. So try to just go through the material. You might find something interesting and that will be staying in your mind. Okay, thank you for joining the class. We'll see you again next week. I'm now ending the meeting.
Thank you, sir.
Okay.
Yes,
Thank you,
sir.
sir.